All right. Ladies and gentlemen, boys and girls, welcome to the 2026 Burton Lecture. Okay, first things first, there are no planned fire alarms. If you hear a fire alarm, run to the exits. Okay? All right. Thanks everyone for coming. It's nice to see a nice healthy turnout. These are an annual long-running seminar series that we have that are a really nice opportunity for us to kind of celebrate outstanding achievements and excellent chemists in our field and share that with the wider community. And I think this is a really great opportunity because chemistry intersects with so many other disciplines. And so in that spirit, I wanted to say a couple things about Harold Burton himself. I found it kind of interesting that he was kind of an early pioneer in spreading chemistry to other fields. So he worked at Guy's, and he developed a course for medical students so that they could learn about the latest advancements in chemistry. And then that was back in the 1930s. And then in 1946, he was appointed to Queen Elizabeth College, which, if you don't know, I didn't, was the women's portion of King's College, which gained independence and then got reintegrated back to King's College, where he taught chemistry to the, brace yourselves, household and social science course. But again, this is in the spirit of spreading chemistry right across disciplines, right? This is the early vein of interdisciplinarity at King's. So maybe from that perspective, you can take heart in this. All right, and then I'm going to hand it over to Saidul, who I think could make a much better introduction than I
Yeah,
did.
thank you. I've just I wrote some notes and I've just deleted a lot of lines because you've covered those bases already. When there's a fire, I run. You shouldn't run. But anyway, so yeah, thank you so much to everyone for being here today. And I should in particular say thank you to Adam Sedgwick, Rob Jefferson and Alex Taylor and all the professional services staff for making sure that today runs smoothly. So you've done an outstanding job. So thank you very much for that. Now, it is my great pleasure to introduce our guest speaker today because I think we're in for a real scientific treat. For many years now, John Sutherland has been at the forefront of origins of life research. He and his group have made many pioneering contributions that have had I would say, a profound impact on our understanding of the chemistry that may have led to the emergence of biology on our early planet. And these contributions are broad in scope. So it could be the conversion of inorganic substances to simple organic molecules, to the assembly of those simple organics into increasingly complex biomolecules, or how the intricate processes such as translation and the format of the genetic code came to be through purely primordial chemistry. Rob touched upon it, but what's particularly significant to point out today is that, chemists are going to have a major role to play if there's going to be a bottom-up recreation of life in a laboratory. And John's contributions are certainly going to have a lasting impact towards that. So let me just conclude with a brief biographical sketch. After completing his undergraduate degree at the University of Oxford, John moved to Harvard as a Kennedy Scholar to work with Professor Jeremy Knowles. He returned to Oxford to carry out his doctoral studies with Professor Jack Baldwin, and after he completed that, he remained at Oxford without choice or force. He was a junior research fellow, and then he moved up as a university lecturer in organic chemistry. He moved to University of Manchester in 1998 to take up the chair of biological chemistry, where his prebiotic chemistry really started to gain prominence. And since 2010, he's been a group leader at the Medical Research Council Laboratory of Molecular Biology in Cambridge. John's been widely recognised through several awards and lectureships, most notably the Royal Society of Chemistry Tilden Prize, the Darwin, that's a ghost, the Darwin Medal, the Stanley Miller Medal from the National Academy of Sciences, and he's also a fellow of the Royal Society. I don't know, I think that's a sign, John. Someone needs to turn the kettle off. So, John, yeah. Thank you so much for being here. It's been a pleasure seeing you again, and the floor is all yours.
Thank you, Leno. Thank you, Rob, for the generous introduction. It's a pleasure to be here, and a pleasure and an honour to give you the Burton lecture for this year. Burton's accomplishments have been alluded to, and it's always a pleasure to remember someone's contributions to this field, particularly at a time when chemistry doesn't have as many friends as perhaps it deserves to have. So thank you, Harold Burton. What I'd like to talk about is, I call this Origins of Life Systems Chemistry. This is really to reflect the fact that whilst a lot of chemistry has been predicated on the idea of A plus B goes to C, life is more than C. And so systems of chemicals, mixtures of chemicals, moderate complexity mixtures of chemicals are what we're aiming at here, not simple transformations of A plus B making C, for example. So that's what I mean by systems chemistry. The problem you have when you start thinking about this is this problem, which is that it's a complex puzzle. How did life start? And there aren't any really blindingly obvious clues. It's akin to trying to do a 1,000-piece jigsaw where all the pieces are white on a white background, okay? And the shapes are pretty similar too, okay? So how do you go about piecing these things together? Well, the first sort of clue you get is that if you think about the way solutions are derived from problems, you want to attack them from as many directions as possible. If you think about this timeline of geochemistry passing through a phase of organic chemistry mediated by the environment called prebiotic chemistry, past this magical moment, then into biology, you can either think about geochemistry and prebiotic chemistry up, what might that chemistry do, what might it create, or you can try and deduce from biology down. And the analogy is that if you try and deduce down from biology and up from geochemistry, it's akin to the situation of building a tunnel. And the tunnel underneath the English Channel was first of all planned by the English and the French independently, both wanting a tunnel to invade the other country. And neither side succeeded in doing that. It was only when they drilled together and met in the middle that they actually solved the problem. So that's a sort of indicator of why we should be adopting this approach. Let me assure you that if we do succeed, we will have a bigger party than these guys had when they finally hooked up underneath the channel. Notice as well that on the English side of the Channel Tunnel, the roof is being held up by a wooden beam. That's kind of worrying. Even worse, on the French side, there is no beam. Okay? So that's what we're going to try and do. We're going to try and deduce things from biology, up from chemistry, and try and meet in the middle. Why don't we start with geochemistry up? Well, most people are familiar with this iconic experiment carried out by Stanley Miller, taking a reduced gas mixture, subjecting it to electric discharge, making tens of thousands of compounds in brown goo, a few of which are actually biologically relevant compounds, isolated in trace amounts, present in trace amounts. But why should we a priori assume that lightning in a reduced atmosphere is the right place to start? It could be all sorts of other scenarios, and all of these other scenarios have been suggested. Do we want to consider a situation where the conditions are static, same temperature, humidity, and so on, or a sequence of different conditions over time? We don't know. In fact, we don't know really enough about the geological scenario to benefit from it. If we did know what the scenario was, if someone had a word in the ear and said it was a submarine vent or it was a drying lagoon, you could then deduce something about the chemistry. But you don't really have enough constraint from geochemistry on its own at the moment to delimit prebiotic chemistry. If we could find some clues about geochemistry, it would assist us, I'm sure. Thinking down from biology, there are two basic approaches. One is to use phylogenetic or sequence comparative analysis to deduce the history of things by comparing sequences. And this gets you back away. It gets you back to Luca. Luca is the last universal common ancestor, a bug or a collection of bugs that basically, from which everything else has derived. Luca was an advanced organism or collective of organisms. In terms of complexity, it's so complex that the origin of life is way beneath this building. It's beneath the tube, it's beneath the deepest thing you can think of. You know? So Luca really doesn't tell you anything about the origin of life. So that approach peters out. A perhaps better biology down approach is to actually either think about a minimal cell or try and experimentally create a minimal cell by taking a simple bug and wiping out everything that appears to be sort of redundant. And either by doing it experimentally or by hypothesizing, you can get down to a minimal cell. And it's got these various components. The problem with that is it's still fairly complex. You need informational molecules if you want to have Darwinian evolution. You need the system to evolve, it's assumed. You need to have building blocks and energy if you want to make things and you want to drive things from a thermodynamic point of view over kinetic barriers. It's easier to make things and replicate things if you have catalysts to speed up and control reaction rates, relative reaction rates. And finally, you can argue that there is no point in genetics or metabolism unless you have compartments. If you innovate, it's as important that you innovate and stop your competitors from stealing that or benefiting from that innovation as it is from you doing it yourself. So you've got to be, altruism is not the name of the game in early evolution. You have to compartmentalize and do things in a selfish way. So that causes a problem because if you start thinking, chemically, if I need to make all of these bits and pieces, nucleotides, amino acids, lipids, a few metabolites as well, if I need to make all of that, that's going to place a lot of demands on the synthetic chemistry. Most people who've done chemistry have made a brown goo in the lab before, Frequently, it's all you get when you do practical organic chemistry. If you try and make that's when you're trying to make one compound that your supervisor is urging you to make. If you're trying to make 50 or 100 compounds in this sort of mixture, you would think you're almost guaranteed to make a brown mess. So there's a caveat here, but it's an assumption. Surely the chemistry would be complicated. Surely you need to break it down into various subsystems. You'd need to make nucleotides separately to the way you make amino acids and so on. And so that led people to try and simplify the problem by saying if you can't imagine chemistry that could make everything, then presumably one of those things came first and then gave rise to the others. And that led to a really awkward split in the field. You would get a group of people, they would segregate towards the four corners of a square room, and then they would trade insults based upon the following analysis. Informational molecules are the most important, so you need nucleic acid. RNA must have come first, the RNA world proponents. Others who said, forget it, RNA far too complicated, you need metabolism. Metabolism wins, metabolism rules, metabolism must have come first, the iron-sulfur world hypothesis, and so on. And others, you can believe it, proteins first and membranes or coacevates first. All unified in the idea that you couldn't make everything in one go, all differing as to what they prefer to think of as coming first, based upon training, bias, whatever, okay? And we kind of got stressed and worried by this because all of these four groups were making good points. Yes, you do need informational molecules. Yes, you do need some sort of driving force, some sort of provision of building blocks. Catalysis will help. Yes, membranes are important. And so we started thinking, well, maybe there's been a wrong assumption here. Maybe it isn't too difficult to make all this chemicals by, in a mixture. Maybe we should actually stop taking assumptions for granted. We should actually start investigating this experimentally. And we were prompted to do that largely by an analysis of the cellular subsystem. So if, for example, you look at the molecule that makes RNA, the molecule that makes an RNA is a molecule made out of protein. The molecule that makes protein is a molecule made of RNA, the ribosome. So separating RNA and proteins, when it's yin and yang with those two, looks like a pretty unnatural thing to do. Membranes, made of lipids, but they're chock full of proteins. One of those proteins in certain membranes is a motor protein which converts a transmembrane proton gradient into the universal currency of energy in the cell. So metabolism is connected to these proteins which are connected to membranes which are part of compartmentalization. Breaking the subsystems apart is dangerous. Metabolism, 1,000 or so reactions, couple of thousand molecules, regulated at the level of transcription and translation. So it's regulated in the other subsystems to control it. The subsystems are all connected. It's like trying to study an arm by cutting an arm off from a body and studying it and then inferring what the function of the arm is. You can do a little bit that way, but you can't really understand the proper function of the arm in isolation from the rest of the body. So the subsystems of biology is not modular, is the basic take-home, according to our opinion anyway. So this led us to start thinking about this chemistry questions. Is it really true you need different chemistries to make the subsystems? Would those chemistries really interfere with each other? If they would, do you need any spatial separation? Is the chemistry Miller-Urey type chemistry that makes thousands or tens of thousands or millions of molecules, including one or two in trace quantities which you want? Or is it a super special chemistry that just makes the ones you want? Okay? If you need to have people in white coats but you can't have them, how does the environment constrain organic chemistry to do the sorts of things that people do in a laboratory? Purifications, sequential additions, and so on. And finally, which components are ancient and which are more recent? We need to tease that out from the system because we want to try and find syntheses of the ones that are ancient and not have to deal with the ones that are more recent. Can we make the whole thing, the whole shooting match in one go, is the question. How are we going to do it? We are going to have a phase funded by the research councils of old where you could do sort of non-applied chemistry, and just explore chemistry for the sake of it and try and find anything that connects the various subsystems, be it mechanism, be it synthesis, anything that relates to subsystems that suggests they could be operated together from a synthetic point of view. We'll then use clues from the chemistry to infer something about geochemistry. If 10 of our reactions need UV light, it's not done in deep submarine vents. If we need, so you could use the results of the chemistry to say something about the geochemistry, then go to your friends in earth and planetary sciences and ask them about other consequences of those environments, feed those into the chemistry, see if it improves the chemistry, take the chemistry results back to your pals in EPS, and see if you can gradually bootstrap your way to a better scenario and a better chemistry. And it's done in the context of a timeline, which is basically the system increasing in complexity and eventually reaching the point at which it transitions from an inanimate state to the animate state. And we think of aliveness as a variable for two reasons. One is because no two people come up with the same definition of what constitutes alive, and having debates gets tedious. NASA's definition doesn't have an ass as being alive, or is it a mule? I don't know, but one or the other. And so having this system here basically transitioning through phases, each one of these innovations, jumps in aliveness, might not get you to the state of being fully alive, but taken collectively, that series of jumps gets you to the point. Okay, so this could be membrane fission, this could be RNA replication, whatever. These are innovations which are then optimized by the system. And we like the idea of there being sort of, systems which are, close to alive and then make the transition to being alive. And we imagine innovations and optimizations just as biology proceeds in a series of innovations and optimizations. What about the building blocks we want to start out with? Well, current biology fixes carbon and nitrogen from atmospheric gases, CO2 and N2. Both of those are amazing feats, okay? Did early biology start out by fixing CO2 and N2? Tricky. Fixing either one in the absence of the other doesn't get you anywhere. So a very simple consideration suggests that a molecule such as HCN, which has carbon and nitrogen, is going to be a very preferred feedstock. And it turns out the oxidation level of nitrogen in HCN is appropriate for biology. The oxidation of carbon is a little bit too high, so you're going to basically need some reduction if you start with HCN, some reduction to get the carbon down to the right oxidation level, which in biology typically centers on the oxidation of carbohydrates. There's a spread, but it's centered on the oxidation level of carbohydrates. So we're going to be looking at HCN because HCN is simple to make in atmospheres through impact chemistry and photochemistry, as I'll show you in a few moments. What sort of chemistry? Well, I alluded to this. Life is not C. Life is not a hugely complex mixture. If you go to your cupboard and you find 3 ultra-processed foods, one of which contains a trace of egg, one's got a trace of oil, one's got a trace of cheese, you cannot make an omelette. Okay? So if you start out with a milliliter mixture with traces of glycine and you can't make sensible peptides from that complex mixture. There's too much stuff that gets in the way. So we want something in the middle here. We think of it by analogy to this sign in a beautiful part of Paris. You've got to have the chemistry sufficiently complicated to get you off in the direction of biological systems that can evolve, but not so complex you end up in a labyrinthine mess. And notice what happens to you if you don't go anywhere, what the birds do to you. We started out in this field, we kind of started out with a bias about RNA being the informational molecule and rapidly became holistic thinkers as opposed to RNA thinkers, but we started out with RNA, so let me start the discussion with RNA. And we viewed RNA, it's a relatively complex chemical structure, a polymer basically of monomers, and we looked at those monomers and just said, well, okay, let's look at those monomers in a very simplistic way based upon our education. You learn about sugars in a carbohydrate chemistry lecture course, nuclear bases in a heterocyclic chemistry course, and phosphate in an inorganic chemistry course if you attend them. And as an organic chemist in Oxford, sadly, I didn't. So I had a lot to learn on the way about inorganic chemistry. I should, of course, have spent more time learning it. But when you see the molecule drawn like that, you immediately think of how to make it. You make it, of course, by taking those components in a preformed manner and attaching them together. Except it doesn't work. Okay? And people have been trying to do that for many, many years, and there are good reasons why it doesn't work. And long story short, we found a way of doing this. Matt is in the audience, I see here. This was Matt's PhD. He showed that if you take half a nuclear base and half a sugar, chemical halves, not mathematical halves, but halves, and mix them together, you get a new product being formed, and that product is formed in the presence of phosphate, which acts as a catalyst, in several ways to actually expedite the formation of that product. You then add the second half of the sugar, you then add the second half of the nuclear base. So essentially what you're doing is you're trying to get to a structure which is 2 rings joined. And what the literature has told you is you can't join these 2 rings, but if you start out with half a ring and the other half of the ring, and then close the first ring and close the second ring, you can make the conjoined structure. And when I explained that in those terms to my wife, she looked at me in a slightly demeaning way and said, "Is that all you do?" And I said, "Well, yes, actually, that is what we do, Anyway, so you can elaborate this into a nucleotide, and the point was there are many ways to synthesize a molecule, not just the one which is subjectively obvious. People criticize this. They say, this is like someone trying to play a golf ball around a golf course and imagining that the golf ball would play itself around the course without the golfer. Okay? It's a bad analogy because he's trying to tell you that the free energy surface that you're playing on is flat like a golf course, okay? But in fact, there's no reason why free energy surfaces have to be flat. They can be really steep. And in fact, if you are a French golfer and you put your golf ball on here, it won't play its way around the course. If you put it here, it will, of its own accord, find its way onto the beach. So if there are low barriers and a thermodynamic driving force, the chemistry, of course, can go on its own. Okay, so the golfing analogy is I think, somewhat fallacious. One of the precursors to the nucleotides, one of the intermediates in that synthesis I've just showed you, happens to be a highly crystalline molecule. It crystallizes as a conglomerate, and on a ferromagnetic surface, which has been magnetized, it can actually be resolved by a phenomenon known as the cis effect. The spins on a surface can be aligned by the magnetic field in a ferromagnetic surface. As the molecule approaches, the dipole moment that's induced in the molecule has a consequential spin dipole, and that spin dipole can be attracted or repelled as singlet or triplet interactions, and those are different for the 2 enantiomers. you can increase the concentration of one enantiomer at the surface and thereby seed the crystallization. And it turns out that that intermediate on magnetite, which is an iron oxide surface which has been discovered, for example, in some of the crater lake basins on Mars and is assumed by geologists to have been present on the early Earth, you can crystallize that intermediate and crystallize it to the point where it's enantiomerically pure. And of course, if you want to make nucleotides, it's best to make them enantiomerically pure, because when you come to polymerize them, you don't face this problem of making diastereomeric mixtures of all the possible isomers that you could get, which would just increase the complexity, make life more difficult. So this work done by Furkan Ozturk, a PhD student at Harvard. We collaborated. We were not major contributors to this. This was majorly done by this young man, Furkan Ozturk, at Harvard, now at Caltech. The next thing that basically came to our attention was basically how to make carbon-carbon bonds. I've told you that the sugars are basically the oxidation level that's basically right for biology. The RNA synthesis starts with half the ribose, and the other half of the ribose, that's a C2 sugar, glycolaldehyde, and a C3 sugar, glyceraldehyde, so you're going to have to make those somehow. If biology is at the oxidation of carbohydrates, why not start there? Well, you don't start there because it's difficult to get two formaldehydes to come together because the two carbons are both similarly charged. So you need to play a trick in modern organic chemistry. You need to reverse the charges to get them to come together. Difficult. But we discovered a way of doing this, which is based upon a very simple reaction where you add a nucleophile to the aldehyde to make the cyanohydrin. And you then discover that you can reduce the cyanohydrin, you can reduce the nitrile, and the reduction is a very facile reduction using one electron chemistry. And that's because if you add an electron and a proton to a nitrile, you generate an immunyl radical. And immunyl radicals are privileged radicals. They're very stabilized. So it's very easy to generate an immunyl radical by an electron and a proton added either together or one after the other. That generates an immunyl radical, picks up a hydrogen atom, becomes an imine, hydrolyzes to make the formaldehyde, reacts with more cyanide to make the cyanohydrin. That cyanohydrin, in the same arrows, can be reduced to make the 2-carbon sugar, and that 2-carbon sugar can be reduced to make the 3-carbon sugar. Now, in this particular experiment, which involved taking hydrogen cyanide and hydrogen sulfide, both extremely toxic molecules, hydrogen sulfide is about 10 times more toxic on the Hegseth scale of toxicity. It's about 10 times worse than hydrogen cyanide. Mix them together, irradiate it with short wavelength UV, and you get these two sugars derivatized as their cyanate derivatives. Don't worry about that. You make these two, but they can't be used from this experiment. They're made as derivatives. But the key thing is you're making them from cyanide using hydrogen sulfide as the stoichiometric reductant, using electrons, and the electrons are persuaded to leave hydrosulfide by short wavelength UV photons photoionizing the HS- to generate electrons which become hydrated. The hydrated electrons reduce the nitrile. Okay? So it's hydrated electron chemistry, aqueous solutions containing electrons, something which was known in the literature but is not widespread use in synthetic organic chemistry. What we've done here in terms of the cartoons I showed before was to do the following. You've essentially taken, you've taken hydrogen cyanide and you've turned it into a carbohydrate, and then you've eventually turned it into the half of the sugar that you need. So this is for those people who are not card-carrying organic chemists. It's homologation, adding one carbon at a time, using a reducing agent to adjust you to the right oxidation level. We call it reductive homologation chemistry. Because it uses cyanide and hydrogen sulfide, we call it cyanosulfidic chemistry, because that's what it is. If you take that reaction and a couple of others, including a major variant of that involving thioamides, you can build up a reaction network, and this network essentially starts with hydrogen cyanide, cyanamide, cyanoacetylene, and most of these reactions basically can be operated in multiple reaction sequences. Okay? Some of them you need to break off and crystallize, some of them you need to dry down and heat, but you've got the bare bones of a reaction network. And the interesting thing about that reaction network is that
it makes a small set of compounds, and those compounds correspond to a subset of the molecules that biology uses. It's as important, or as interesting rather, for the compounds it doesn't make as for the ones that it does make. So it doesn't make a huge mess, it just makes a few compounds, and those are the compounds it makes. Okay, so that enables us to answer a couple of questions. Are completely different chemistries needed to make the different subsystems? No. Is spatial separation of parts in the network necessary? Yes, almost certainly. We don't know exactly the details, but we can't envisage doing the whole thing in one go. So we're going to have to break it down somehow into separate streams or whatever, as you'll see. It looks like intrinsically favoured chemistry making a few products rather than indiscriminate chemistry making loads of stuff, including traces of what you want. So intrinsic chemistry. Where do the building blocks come from? This is the atmosphere on primitive Earth. Outgassing was thought to have been similar to outgassing now. It's basically a consequence of the redox state of the mantle, which hasn't changed significantly over time. And then superficial hydrothermalism or volcanism can release these outgassing components, typically fairly oxidizing atmosphere, but it can be converted into a reducing atmosphere by a giant impact. The impactor core is iron or iron-nickel alloy. You disperse that in the atmosphere, you reduce water to hydrogen, the hydrogen reacts with CO2, and you convert the CO2 down to methane through CO. If you allow an atmosphere, a reduced atmosphere like that, to undergo a tropospheric photochemistry, you generate large amounts of hydrogen cyanide and cyanoacetylene. So these are the ways in which the building blocks can be made, and they can be brought down to Earth by dissolving in water. So Henry's law, dissolution of HCN, harvest them, for example, as ferrocyanide. Iron reacts with cyanide to make ferrocyanide. Hydrogen sulfide can precipitate as transition metal sulfides. Phosphate accumulation can be allowed by precipitation of calcium as its carbonate, which stops calcium precipitating the phosphate. So these are ideas developed with, card-carrying geologists, John Grotzinger, David Catling. And the idea is that you can then recover the cyanide from the ferrocyanide, for example, by heating. And the sorts of cyanide derivatives you get back after heating are contingent on the nature of the cations associated with the ferrocyanide. Sodium potassium cyanide give you back, ferrocyanide give you back sodium potassium cyanide. Calcium ferrocyanide gives you calcium cyanamide. Magnesium ferrocyanide gives you magnesium nitride. So you get a set of building blocks in the dry state, which you can then dissolve in water to any concentration you want. And by flowing water across a thermally metamorphosed surface, you can imagine doing chemistry in streams under the influence of solar radiation, and then those streams intersecting, and you get mixing of the chemistry as you proceed. Because you're in a fluvial or lacustrine setting, if it dries out, you can crystallize compounds. So the crystallinity of that intermediate requires a crystallization step. Again, you're not going to do that in a deep ocean environment. Okay, so there are multiple clues here that you should be doing this on the surface. Sequential chemistry, well, if you have a solution containing sodium and potassium cyanide and it flows over a metal sulfide, it will dissolve the metal sulfide, if it's iron or copper, for example, generate this corresponding ferrocyanide or copper cyanide complex, and liberate hydrogen sulfide as its conjugate base, if the pH is above 7. So you can generate streams which contain hydrogen cyanide and hydrogen sulfide pretty easily in this way. It is thought. Now, here's the fun bit. We saw that at the beginning that the analysis said that RNA and proteins looks like they've shared a common origin. If they have shared a common origin, in biology they're really tightly associated, RNA and proteins. If nucleotides and amino acids are made by similar chemistry, then surely RNA and proteins were born together and have been living together all their lives. So we should start looking for some connections between RNA chemistry and protein chemistry. And we did this, first of all, by looking at the genetic code. So for those of you who are pure chemists, the genetic code is the relationship between the language of nucleic acids, the sequence of nucleic acids, and the language of peptides through the sequence of amino acids in a peptide chain, as dictated by the genetic code determined 50, 60 years ago. Now, the genetic code basically enables the information in the process of translation to be decoded. The coding step, if you like, is where amino-acyl tRNA synthetases attach amino acids to the dial at the 3' overhang of a tRNA molecule, and they do that having recognized identity determinants normally elsewhere on the molecule which tell the enzyme it's the right tRNA to attach this amino acid to. Once the amino acid is attached, the sequence of the anticodon is then very easily connected to the sequence of the messenger RNA through Watson-Crick pairing as controlled by the decoding center, the 30S subunit, at least of the bacterial ribosome. So the genetic code is basically is written by the enzymes and read by the ribosome. And how that whole process arose is difficult. And I love this. I guess I can actually get away with this slide here because, you know, you guys don't probably hero worship Crick to the extent that other people do. Okay, but here's a paper where the idea apparently in this paper was put forward by this author, and these three characters managed to get their name on the paper. I find that quite amusing. But in the process of doing that, this guy wrote the first sentence, which is this sentence here: the origin of protein synthesis is a notoriously difficult problem. No kidding. I mean, you write that in a paper nowadays, and referee number 2 would say, well, they'd come up with something, wouldn't they? No, but that's great. What a great sentence, because, you know, it says what it is. But notice this: we do not mean by this the formation of random polypeptides, but the origin of the synthesis of polypeptides directed, however crudely, by a nucleic acid template, and of such a nature that it could evolve by steps into the present genetic code. In other words, not just any peptide synthesis, not random peptide synthesis, not RNA-controlled peptide synthesis that can't morph into what we now see, but it's got to be RNA-controlled peptide synthesis where you can see a trajectory to take that chemistry to the chemistry now used by biology. So Crick is being pretty constraining in what he thinks we should have. And I only put him up because I think he expresses an opinion which I personally like. You know, we shouldn't care if a particular person says something, we should care what we think, right? But I just happen to agree with this. I think that's absolutely key to this process. Let's look at the genetic code again. The genetic code has got two sorts of boxes which can be distinguished. One like this one, with a bold around it, where the nature of the third base of the codon doesn't influence the nature of the amino acid that's coded. It's called a family box, or fourfold degenerate, fourfold synonymous box. And right next to it is a box where changing the nature of the third base of the codon changes the nature of the amino acid that's coded. It's called a split box because it's split. So about half the boxes are family boxes, about half of them are split. Where do the amino acids that we make sit? Well, those are the amino acids we make in red. They sit precisely on the family boxes, which is, you know, is really to us kind of, you know, a clue, because it's saying that there is something special about family boxes, and you've made these 8 amino acids, and they sit on those family boxes. What's the connection? Well, the family boxes, if you have a quick look at what they are, they're boxes where the first two bases, if you look at the first two positions, it's either 1 or 2 G's or C's in the first position. If it's only 1 G or C in the first position, the second position is a pyrimidine. So here you've got 1 G or C in the first position, second position is a pyrimidine. Here you've got 1 G or C in the first position, but the second base is a purine. So this is a split box, this is a family box. Okay, so split boxes, family boxes. Number of GCs hints that it's something to do with the strength of the base pairing of the codon to the anticodon. GCs form stronger base pairs than A's and U's. So is there something about base pairing strength of codon to anticodon that might be important here? You know, if there was, it would make sense, because then you would find that, you know, if the codon-anticodon binding was part of the binding that was responsible for putting 2 amino-acyl tRNAs into proximity to enable transpeptidation, then it would make sense that you assign your first amino acids to the RNAs which have the tightest intrinsic binding to the message. So you'd put them onto family boxes. The other reason why family boxes are actually tighter than other boxes is because if the second base of the codon is a pyrimidine, the second base of the anticodon is a purine, and if there's a purine here, you can get an extra hydrogen bond from this uridines 2' hydroxyl to N7 of the purine. That intra-loop hydrogen bond rigidifies the loop and reduces the entropic penalty that's paid when you bind a loop to a piece of message. So having a pyrimidine here gives you an extra hydrogen bond. So family boxes looks like it's just base pairing strength and loop tightness, which basically is expressed through base pairing strength. If you measure, we synthesized all of the family box and split box anticodon loops by chemical synthesis and looked at their binding to messenger RNA by biolayer interferometry and isothermal titration calorimetry, but this is the BLI data. Here are two family boxes, one and then the other separately, and they bind with low micromolar affinity. The split boxes bind 50 to 100 times weaker. So split box binding, a lot weaker. Now, in the literature, it's claimed that you can't bind two anticodon stem loops or two tRNAs to message without the ribosome. But that was measured using a split box tRNA for technical reasons. So we wondered if, with family boxes, we might be able to get two tRNAs to bind to message on their own without the ribosome. And it turns out you can. Here's one pair of family boxes, you bind one, and then you sequentially now bind the second, and you can do it either way around. So you can bind two family boxes to message. Here's another two family boxes you can bind to message. We now have NMR solution structures of these anticodon stem loops bound to message in collaboration with Victoria de Souza at Harvard, an NMR spectroscopist. You can see the message is kinked as it is in the ribosome, the two anticodon stem loops bound, but there's a twist to the structure. And that twist, we think, is resolved if you have the full-length tRNAs, because we have biophysical data that suggests with the two full-length tRNAs, they're bound as they are in the ribosome. Furthermore, if we take a short piece of the ribosome, which corresponds to what the ribosome now uses at the core of the decoding center, and we make that just as a short oligonucleotide, 40-odd residue oligonucleotide, we can take that decoding center and we can look at the binding of anticodon stem loops. Here's one binding, here's the second binding. The first and second binding have similar strengths as adjudged by the ratio of the on and off rates. If we add this synthetic 42 residue oligonucleotide decoding center, you can see that the second binding, the on rate for the second binding is substantially increased, which reflects a tighter binding. Okay? So it's the on rate that's altered. So what's happening with the family boxes is that they bind tight. They bind even tighter if you have this minimal decoding center present. So it's beginning to look like the 30S subunit of the ribosome comes first, initially without the decoding center to enable transpeptidation, and then with the decoding center. Beginning to look like means I'm speculating. It means that's where we're planning to go. We haven't by any means proved that yet, but that's the direction of travel. We also find that the decoding center does what it does in the ribosome. It basically makes the binding of cognate, which is the right anticodon, to cognate codon binding tighter. So all of these cognate bindings are increased by the decoding center. And if I look at mismatches, mismatches are penalized in much the same way that they are in the ribosome itself. So mismatches at the first position are completely destabilized in these cases, heavily destabilized here. Mismatches in the second position, similarly destabilized. So the 42 residue decoding center is increasing the fidelity of the binding of the short oligos to the anticodon stem loops, as it does in the ribosome. So very briefly then, what about the other step, the writing of the genetic code? Well, that requires amino acylation. And the chemical amino acylation of RNA has a selectivity problem. You've got a variety of different nucleophiles on RNA, all of which can be potentially undergo reaction with an acylating agent. And early studies on the amino acylation of dinucleotides as basically proxies for longer RNAs demonstrated that if you take something which is good enough to acylate a diol in water, it also acylates the internal 2' hydroxyl. So there's not a big element of selectivity. And we reasoned that if you want to do this chemistry in the presence of, for example, concurrent RNA replication chemistry, all of this sort of adventitious amino acylation might be deleterious, so we should explore chemistry which can basically do the amino acylation more selectively. And we were driven again to an idea about how to do it more selectively by thinking about how amino acyl tRNA synthetases do it. So the enzymes that do this react amino acids with ATP, make a mixed anhydride intermediate, and then use that mixed anhydride to acylate the 3' overhang of a cognate tRNA acceptor stem. So our idea was simply to take this amino acyl adenylate and extend off this 3' hydroxyl, extend a chain that could bind to the corresponding cognate acceptor stem strand. So we made, using synthetic chemistry, not prebiotic chemistry, we made mixed anhydrides here, and we looked at the transfer of amino acids across from the phosphate to the alcohol, and that chemistry takes place. It's to a degree, it's stereoselective. You know, the stereoselectivity is of the order of 10 to 1 with the right sequences for the right relationship between amino acids and RNA. And it's to a degree chemoselective. And we looked at the chemoselectivity by varying, we kept a certain overhang sequence, and we were getting clues that there might be some selectivity in the chemoselectivity of the process in the terminal trinucleotide of the molecule. So we used a process where we could do a high throughput analysis. We took random sequence libraries, amino acylated, and then had a chemical trick where we could basically sequence only those molecules which basically underwent successful amino acylation. And when we did this, we found that there were sequence preferences in this terminal trinucleotide which were different for the different amino acids. Notice that valine and leucine are not that different. Okay, so there's a clue that the chemistry might be picking up something to do with the side chain. And we went, you know, you can't just use that sort of analysis to sort of conclude anything hard and fast, so we went and synthesized these things and used HPLC and triplicate and so on to actually analyze this, and we found, for example, here's a sequence, a trinucleotide sequence where L-valine is acylating faster than D-valine, but also faster than other amino acids. The differences here are not dramatic. They're very small differences, but I think imagining how you get coding or adventitious coding using trinucleotides, I don't think you're ever going to get very significant coding unless you start evolving bigger RNA structures to do this. And the idea here is that this precedes biology trying to improve it. This is what you start out with. So if there's any coding to start with, the feeling is that it might be easier to improve that rather than to develop that selectivity de novo. So you change the sequence and you basically change the nature of the amino acid that's preferred. For example, here, valine not liked at all. Here, valine liked but not liked as much as leucine. We do not know why there is that selectivity. We've looked on numerous occasions to try and get sort of some sort of structural analysis that will tell us why that preference is being expressed. We do not have an answer. Probably NMR is going to be our only way of doing it, and we haven't invested in doing that yet. We think this is related to what was proposed by Christian de Duve, based upon a large amount of data in the literature, that frequently the identity determinants that the enzymes use to recognize the identity of the tRNA don't just reside in the anticodons stem, which is an obvious place to have them, but a lot of them cluster in the last couple of bases, last 3 bases actually, of the acceptor stem. This was referred to by de Duve as a second genetic code. It's curious because he was actually thinking of a second genetic code which actually came before the primary genetic code. And so this is basically, it's amusing because it kind of is a way of having a compromise between the views expressed by Crick and those expressed by Verza about the conflicting sort of ideas about how you might assign the first amino acids. Kind of says there might be, you know, an element of the frozen accident hypothesis here and an element of what's called the stereochemical theory where nucleotides recognize amino acids and vice versa. And if you look in existing biology. You find here is the clustering of the identity determinants. These are the sequences that basically the enzymes recognize to identify whether or not it's the right tRNA. And they cluster in the anticodon loop, and they cluster in the acceptor stem. So we think this might being the operative word, reflect an earlier second genetic code in the acceptor stem. Last but not least, transpeptidation. Well, we are nowhere with transpeptidation yet, except to say we have a better idea about the difficulty of the problem. Let me cut to the chase here. If you do model studies, and these are an extension of model studies which were first published by Leslie Orgel, what you're doing is you're looking at the reaction of an amino acyl ester on one tRNA with an amino acyl ester on another tRNA, and you're doing that, in reality, in biology, one of those amino acyl esters is acylated on nitrogen, but it's a simpler model study to look at the free amino acyl compounds and look and see what the rate of transpeptidation is as a fundamental chemical reaction. And it turns out it's a really bad reaction for a couple of reasons, and it's really bad because of competing hydrolysis. So these amino acyl esters are very easily hydrolyzed. And if you want to try and make a peptide by bringing them together, you have to bring them together such that the concentration of the two of them to react is higher, or effectively gives you an effective reaction relative to 55 molar water, or actually the hydroxide, whatever the concentration of hydroxide is in the solution at the pH you're working at, which is typically somewhere 7 and a little bit above. So hydrolysis is a pseudo second order reaction. It's a very facile reaction, and the reaction with the amine really requires that you deprotonate the tetrahedral intermediate so that it can break down to give you the amide product. If you don't deprotonate that nitrogen, it bounces back to give the starting material. So the transpeptidation is bad chemistry. It's third order chemistry. Hydrolysis is much more facile. It's second order chemistry. So you're trying to get third order to go against second order. It's difficult to do. So even at 600 millimolar in the one and a few millimolar in the other, you still get a whole load of hydrolysis. Okay, with things like glycine, your transpeptidation or your aminolysis yields are very low. But serine is much better than other amino acids. And the reason that serine is much better, long story short, the reason that serine is better is because serine doesn't react on nitrogen to start with. It reacts on oxygen, and it makes an intermediate ester, and that ester is then transformed by intramolecular migration of the acyl group onto nitrogen to give what looks like the product of transpeptidation, but is in fact an O to N rearrangement product. So this is really curious because it suggests to us that, you need to get proximity to do transpeptidation. And even if you get of the order of 1 molar effective molarities between these termini, you're still only really going to get peptides formed if you have serine as the nucleophile. To make all the other peptides, you're going to have to get better proximity. You're going to have to start desolvating the reaction center, we think. And if that's the case, then it raises this question, if the ribosome is making X serine peptides and is then acquiring the ability to make others, what's the chance that X serine peptides are going to be functionally useful? Well, they might be, but there's another possibility, and the other possibility is that in fact, it's not the peptide products of the ribosome that are important, it's the fact that you've taken a blocking group off the end of an RNA. And if you're doing concurrent RNA replication chemistry that needs that diol to react with a phosphate to make a phosphodiester bond, having amino acids or acyl amino acids on the end of your diols will clog that replication chemistry up. So you have an evolutionary incentive to deacylate those termini to improve RNA replication chemistry. So here the idea is that maybe there's a selection for a function, and selecting for that deblocking function then gets you to the point where biology is ready to make a small change in the function which is being selected, an exaptation step, such as that exaptation that's thought to have occurred in the evolution of flight in birds. And that then sets the RNA up to do proper transpeptidation. So I hope what I've showed you is that, yeah, you can start off thinking about this origin of life problem and you encounter all sorts of conceptual difficulties. You start doing some synthetic chemistry and you start getting some clues from the synthetic chemistry. And those clues don't kind of stop at an imaginary boundary between chemistry and biology. They permeate deep into biology, all the way through to the central dogma of biology and the assignment of amino acids to the genetic code. So biology, half of the early part of biology looks chemically deterministic. If you repeated it, you get the same core attributes. Later amino acids might well be assigned on the basis of contingency. They might be different if you reran the tape. But chemistry is reaching into biology. So chemistry is an important topic to study for the origin of life. Where are we in terms of overall progress? Well, we have, this is just stylized. I couldn't get a jigsaw puzzle on the web where we'd put in fewer white pieces, so this is probably over-optimistic. But we've put in some of the community has put in some of these white pieces over the years. And it's like any puzzle. I think as you put more pieces in, it kind of gets easier to put the final pieces in. So the hope is that the end is in sight. Finally, of course, I'd like to thank those people in my group. The ones in bold are the ones who have a life sentence in my lab, full-time employees of the Medical Research Council. The others have had only a short sentence, so several of these people have escaped. Others are still with us. Intellectually assisted by these characters primarily, and additionally these characters, some of whom I've referred to, and funded by philanthropic organizations in the US and the Medical Research Council, both groups of which take a view that long-term funding of speculative research has a place in the portfolio of research funding in chemistry. So my thanks to all of them. My thanks to you for the invitation to come here and for your attention.
Any questions? Manuel? Sorry, Manuel.
Fantastic talk, and it was really nice to see how the different approaches converged. What was striking in your original amino acid alphabet is the absence of most of the catalytically proficient amino acid side chains. So do you think they were just not important at that time, or were other functional groups carrying out the catalytic functions? And maybe a follow-up question then, how would you catalyze some of the reactions in the cyanosulfur metabolism if you could?
Okay, so there's a couple of questions there. If you like, the quotes more useful amino acids. Well, this is one or two criteria, synthesis and family box assignment, two criteria according to which those amino acids are arguably more ancient. There are other clues, if you look at the phylogeny of amino-acyl tRNA synthetases, for example, that suggest, the AAT RNA synthetases partition into two classes, 1 and 2, and those sub-partition each one of them to A, B, and C. And A look older than B, look older than C. Most of those newer amino acids are in groups B and C. Ones that we would say are newer are actually phylogenetically newer. Split boxes are not just different to family box on the basis of codon-anticodon binding. Decoding of split boxes relies upon nuclear-based modification in the tRNAs. Post-transcriptional modification now affected by enzymes. Some have argued that it could have been affected by RNA catalysts, but I'll tell you what couldn't have worked, which is what some people have proposed, is that you actually have bases, for example, T6 adenosine, which has got a 3-enol carbamyl group on N6 of adenosine, which actually sort of sits, position 37 of the anticodon loop, presents a bigger aromatic stacking surface to stack onto the weaker codon-anticodon duplexes to stabilize them. So that increase in aromatic stacking is what stabilizes the weaker ones. That modification to the adenosine blocks its Watson-Crick base pairing, so that modification couldn't be reproducibly incorporated by, you know, sequential cycles of replication. It would have to be introduced randomly at each cycle, at just that position and not elsewhere. Elsewhere it would block Watson-Crick function. So there's quite a strong argument that says that nuclear-based modification needed to accurately decode split boxes is a late arrival. So I think if you add all of those things together, it's by no means proof. It's by no means proof. Second part of your question, how do you catalyze the other reactions? Well, the idea is that you don't just have short peptides in the mix, you have short RNAs in the mix as well. They potentially provide scaffolding to the peptides, which is a concept which I think several groups are investigating. We're investigating it, and I know several others are investigating that too. The idea is like the photograph of the two wounded World War soldiers leaning on each other as they walk down the street. Either one on their own would sadly fall over, but together they can just about make it along the road. So can short peptides and RNA achieve something together that they couldn't achieve on their own? And I think the third point is that, you know, some of the chemistry underlying cofactor synthesis is highly predisposed. In other words, it's kind of a distinct possibility that some of the cofactors could have been produced by this sort of cyanosulfidic chemistry. And a lot of the chemistry that's catalyzed by enzymes that use cofactors will occur in the absence of the enzyme, in the presence of the cofactor, albeit to a lower degree. So a combination of RNA-assisted peptide chemistry and cofactor or nascent cofactor chemistry, coupled with the fact that there's a difference between the chemistry that you're using in extant biology to make things and the chemistry you use in prebiotic chemistry. Extant biology deliberately uses reactions that are bad in the absence of catalysts. Because if they weren't, you couldn't switch them off. And if all of metabolism was permanently switched on, you know, you'd go out with a bang, right? Interesting experience, but everything would be going at full tilt, you couldn't turn it off. So to be able to turn it off, you've got to down-regulate the catalyst, which means the background reaction has got to be bad. So biology has deliberately chosen bad reactions. Prebiotic chemistry has got to occur by and large without catalysts, you know, not strictly, but by and large. So to a certain extent, the reactions that you're relying on early on are going to be less reliant upon the catalyst that you need later on. So I think that's the rough outline of the answer to you. Yeah.
Great. Thanks very much.
Hi, John. Thanks a lot for your great talks. I want to ask you about the cis effect that you picture there, just to know your perspective, because it's hard to me to get that. I work on the cis area, but not in the chemistry side of things. So you have a magnetic surface, and then you start with some chemicals that are not chiral, and then to me, when the chirality starts, is the reaction happening nearby the magnetic surface and the effect of the magnetic field during the reaction, or is something else there that I'm really missing?
So the key molecule is chiral, but it's not enantiomerically pure, it's racemic, because it's been produced, you know, without any chiral influence. So it's produced in racemic form. And the idea is that if that racemic material has the property of being a conglomerate, conglomerates crystallize as separate enantiomers, as opposed to mixtures. So if you have a conglomerate, basically, then if you increase the concentration of one enantiomer at the surface, you can seed the crystallization. As long as the crystallization doesn't go to completion, so you don't crystallize all the material in solution, that percentage or that fraction of material which is crystallized is enantiomerically enriched because of the cis effect interaction. So it is chiral. It's a conglomerate crystallization, and that's a requirement, and it's the interaction of the different enantiomers, the two different enantiomers.
But then the chirality is there already, so the chiral molecule is there. It's just the interaction, the specific interaction with one isomer of the magnetic surface that induces.
Is it?
So the chiral molecule has to be, you need to start with something chiral. It's just a racemic, but you need...
Yeah. So we're not doing a chiral induction, which is where we take an achiral molecule and turn it into a chiral product with enantioselection. We're taking a racemate and resolving. Yeah. So we did try with Furkan. He came to our lab, and we were trying to use spin polarized electrons that are produced by filtration, you know, using molecules sitting on a surface, for example, using the cis effect to give us spin polarization. We were trying to use spin polarization, because we're using hydrated electrons to do our reduction chemistry, we thought, can we use spin polarized electrons to do that? The problem is the spin polarization decays too rapidly before the electrons have had a chance to encounter the substrates. So the spin polarization decays very rapidly. Now, I'm not an expert, so this is what the physicists have told me. That the reason we didn't see any induction was because of that. We then discovered basically that you could, in principle, do this by crystallization. We knew that REO was a conglomerate. We knew basically that it's highly crystalline, so we suggested to Furkan that he try the crystallization on magnesite. For what it's worth, we now actually prefer the idea of using greigite, Fe3S4, because if we use ferrocyanide and we've got hydrogen sulfide present, there are ways in which you can photoequate the ferrocyanide and eventually produce iron sulfide. And you can photooxidize iron sulfide to make greigite. So we kind of think an inorganic byproduct of the organic synthesis might be greigite, and if that settles out from the reaction mixture onto the floor of the, whatever it is, the basin or the puddle, then you'll end up with a ferromagnetic surface, freshly prepared, as a result of the accompanying inorganic chemistry. So greigite, I personally think greigite is a better bet than magnetite.
Thank you.
Sorry, I'm just from an inorganic chemist background. Some of it's been a bit above me, but
Did you say you're just an inorganic chemist?
well, in the
No,
background.
on the contrary.
Well, I did do organic metallic chemistry for
Okay.
a year, so I did do some organic chemistry. I was just interested to know. I was going to ask that same question just about chirality. It's, if I've got it right, it's L amino acids and D sugars.
Yeah.
So, are you going to be doing stuff with D sugars as well too?
Yes. So once you've got the one fixed, the rest is a relative stereochemical argument. So if you start with, let's say you start with L sugars and D amino acids, you'll have exactly the same but mirror image opposite if you start the other way around with both. So it's a relative, the chemistry that follows, the stereochemistry that follows is so-called relative stereochemistry. You're relating the new stereochemistry to the existing stereochemistry. And you get the right relative, I mean, it's a 1 in 2 chance, right? So it's not dramatic. But you get the right relative stereochemical connection when you amino acylate the tRNA by that transfer chemistry. If you've got a dRNA molecule, you transfer L amino acids faster than you transfer D amino acids. And that which isn't transferred gets hydrolyzed. So you end up with a chirally selective amino acylation reaction. And it's sufficiently chirally selective that you could get chiral enrichment of, peptides comprising several residues before you ended up with, really complex diastereomeric mixtures. So it's arguably a workable enantiomer enrichment that you would get for that relative stereochemistry. If you flip the stereochemistry around and it started out with L sugars, it would be D amino acids. So it's a relative stereochemistry. It would be the same relative in the mirror.
And could some of the RNA come from space in meteors?
Well, the argument behind that, I mean, I don't know if you've seen the film Life of Brian, where Brian is being pursued by a crowd, and the crowd is keen for a sign. Give us a sign. And Brian's shoe comes off, and someone picks it up and says, a sign, look, a sign, okay? And the point is, it's very easy to interpret one data point and interpret it as a sign. So when the astrochemists tell you that there's on a meteorite or something there is some glycine, or there's some adenine nuclear base in whatever, imagine what happens when that hurtles down to Earth and is impacted. If any of it escapes atomization, then it's there. We know what the composition of the organic inventory on Murchison is. It's hundreds of thousands of compounds. So anything that survives is in one hell of a mixture. And the argument is it's not going to contribute, because if you start trying to assemble building blocks into higher order structures, if you've got 1,000 amines alongside the amine you're trying to acylate, you're going to make 1,000 byproducts. And at the next step, you'll make 1,000 for each one of those. You'll make 1,000,000 byproducts. So these mixtures that you get from space, to say glycine in a meteorite, holy cow, it's a sign, I think that's a Life of Brian moment.
So it's not likely that these things will fall.
In my opinion, it's not likely. If you ask an astrochemist, you might get a different version of the story. You should ask an astrochemist.
Okay, so we've run out of time.
Yeah.
So everyone give John a round of applause.